Okay, everyone, welcome back to Cambridge AS and A-Level Biology. This is part 2 of the May June 2021 paper 4 variant 1 solutions. So if you're starting here, or maybe this is the video you found first, please go and watch part 1. I deliberately broke the videos into 2 because paper 4 is quite a long paper, and I'm kind of worried that when the videos are maybe like an hour long, some of you don't follow through to the end, because I get comments that keep asking me questions like questions I've already addressed in the videos, basically, which sort of lets me know that you did not watch the video to the end. So if you haven't watched the first part, please do so. What I will reiterate here is that your paper 4 is a A2 paper, and it covers chapters 12 to 19. So by all means, make sure you study that. Most of the time, it is the first paper students have to write, and it is also the longest paper. It's for 100 marks, and you have 2 hours to write it. So it requires you to know a lot of detail and how to apply the information you have learned from chapters 12 to chapter 19. Please don't ask me questions like what chapters are important of all these chapters. All of them are important. You should study every single chapter because you will encounter questions from every single chapter. So don't play any, mini, money, more here. Just get studying, make sure you study everything that you need to know, and be well prepared for your exams. So let's get into part 2 of this paper. So part 2 starts at number 5. The last one stopped at number 4. So if you haven't watched the last one, please make sure you do so. Let's see what this says. It says here, nerves consist of bundles of different types of neurons. Figure 5.1 shows the mean transmission speed of impulses for 3 types of sensory neuron. And it's A, I think that's like A, is that omega? No, that's not omega. A, that's sine, that's delta. I'm not sure. I can't remember my numerals anymore. I only remember them in English now. And then there's A, beta. Anyway, these are the 3 different neurons there. And then it says impulses from pain receptors in the skin are sent along sensory neurons C and that one. Sensory neuron C and that synapse in the spinal cord with relay neurons known as projection neurons. Projection neurons send impulses to the part of the brain that perceives pain. Impulses from touch receptors in the skin pass along sensory neuron A beta, which can also synapse to the projection neurons in the spinal cord. Sensory neurons A and A beta are myelinated, sensory neurons C are unmyelinated. So first things first, I would maybe like take note of the fact that they're telling you here that these are myelinated neurons, because you know that the myelinated neurons are the ones that have the myelin sheaths around them, and because of that sheath, they're able to transmit neurons impulses a lot faster. And just in case you're wondering where this is from, this is chapter 15. And if you're wondering and you're writing in October, November, I would be very, very worried. Please study hard. October, November is literally around the corner. So let's see what the question says. Describe the results shown in figure 5.1 and suggest explanations. So again, I am going to go in here and tell you whenever you are asked to describe, it is telling you to show or to put in words the trend that you see. That's what describe means. So when you look at this graph, what is the trend that you see? Here is the axon diameter, and this is the mean transmission of the impulse. The trend you see is that C has the lowest axon diameter and also the lowest mean transmission of impulse, while A beta has the highest one, highest axon diameter and the highest mean transmission, mean speed of transmission. So those are the things that you can speak about. So you can then say, okay, based on this trend, what I see is that the higher the transmission, so higher transmission, higher diameter rather of axon. I'm just going to write it as diameter of the axon, the higher the speed of transmission, or the mean transmission speed. Higher the mean transmission speed. Writing this way is actually quite uncomfortable, writing with a mouse. I'm not an artist by any stretch of the imagination. So mean speed, okay? So I hope you get the gist. That's the trend that we can see. And obviously then you can go back into the question and say, okay, well, we were also told here that these 2 are myelinated neurons, right? And they are the ones that have the highest speed of transmission. So you can then say perhaps the reason why A I don't know what that sign is. For some reason, I've forgotten it. And A beta, the reason why they have such high transmission speeds is because they are myelinated compared to C, which is not a myelinated neuron. And you can also then refer to the fact that because they are myelinated, they're able to conduct what we call saltatory conduction. Okay, and that will give you full marks. Saltatory, I think that's an A over here. Yes, saltatory, because that looked weird. So you can then speak about saltatory conduction, talk about the presence of the myelin sheath, and that should give you full answers. Okay, then 2 says, gently rubbing a damaged area of skin can reduce the perception of pain. Suggest an explanation for this. So I guess something here it says suggest. So this is not based on fact, but it has to be deduced from fact, if that makes sense. So if you're rubbing a damaged area of your skin, it can indeed reduce the perception of pain because it will maybe transmit the impulses of you putting your hand on your skin much faster than the pain itself. So like for example, if you, how do I explain this? I'm trying to explain this in a way that you would understand. So let's say, for example, you hit your head. Not a great example, but let's assume you hit your head. Chances are that you will feel some level of pain, or maybe you have like something that feels like a splitting headache. But let's say that while you're feeling that headache, you take your hand and you touch your head. What you will suddenly start to feel is the temperature of your palm. So you might be like, oh, my hand is cold, or my hand is warm, or whatever it is that you're feeling. And that's because the touch sense tends to be transmitted a lot faster than the pain itself. So that might be a reason why rubbing an area of the skin can reduce the perception of pain. All right, let's look at B. Over here, it says back pain in humans can be reduced by using a transcutaneous electrical nerve stimulation machine. So that's a TENS machine. A TENS machine uses electrical impulses to stimulate nerve endings near the site of the pain. It is thought that TENS triggers the release of natural painkillers called endorphins at synapses. And then they show you a synapse, okay? So if you again have watched the videos on chapter 15, this is not foreign to you. With a synapse, you have what you call the presynaptic neuron and you have the postsynaptic neuron. Over here, this is an acetylcholine receptor, this is the endorphin receptor over here, and based on what I am seeing here, I can see that the endorphin receptors are here. They are not within the synapse, like they're not anywhere in the synaptic cleft. So this region here would be the synaptic cleft where I've drawn the arrow. The endorphin receptors they seem to be on the side of the presynaptic neuron. So what I believe would happen, and this is the question that they ask over here. They say, how would suggest and explain how endorphins may act to reduce pain. Now remember that pain is some sort of nerve impulse itself. So it means that whenever a person is in pain, let me just erase here so that I can explain a little clearly without anything in the way. So it means that when there's pain, and you can look at the arrow, you don't even need to think too much. So over here it says impulses from area of painful impulses from area of painful back muscles, not black muscles, back muscles. So the impulses will go here through the presynaptic neuron. Okay, when they get to the presynaptic neuron, remember how synapses work. They will stimulate the opening of calcium gated channels, and that calcium gated channel will result in calcium flowing into the presynaptic neuron, which will then cause the release of these vesicles over here. These vesicles are containing acetylcholine, or they're carrying acetylcholine, and so they would then move into the synaptic cleft and bind to the receptors. So acetylcholine will bind to the receptors over here. So the vesicles will fuse with the presynaptic membrane, release acetylcholine, it binds to the receptors on the postsynaptic membrane, and obviously that sends a message to the brain to say there's pain happening here. But in this case, we're saying endorphins might be able to stop that. And how would they then do that based on what we can see on this image? So I'm erasing again. Let's look at where our endorphins are. So look here, it says impulse from area stimulated by the TENS machine. So the TENS machine is helping with the endorphins. The TENS machine will then send an impulse into this neuron over here that has vesicles containing endorphins. The endorphins will go out and they will then bind to the receptors over here on the presynaptic membrane. If the endorphins bind to the presynaptic membrane, their point is to prevent pain from being felt because they are natural painkillers. So in order for them to prevent pain from being felt, it means they need to stop this whole process that happens here, where acetylcholine will move through the synaptic cleft and bind to receptors on the postsynaptic neuron. So what you can then say in this case is that the endorphins will bind to the endorphin receptors, obviously. And what they will then do by binding to those receptors is that they will stop the calcium-gated channels in the presynaptic membrane from opening, or the presynaptic neuron or presynaptic knob. Totally dependent on you how you put that, but it's always a good idea to use the description that's put on the image. They will stop the calcium-gated channels on the presynaptic knob from opening. If the calcium-gated channels don't open, it means that the vesicles containing acetylcholine will not move to the membrane and fuse with it and release acetylcholine. So here is the steps then. I'm not going to write the whole thing down, all right? I hope that you are taking notes as I say it, because writing it down will just make this video unnecessarily long. So here we go. Endorphins will bind to endorphin receptors. They will prevent the release or the opening of the calcium-gated channels. There would be no release of calcium, and as a result of that, the vesicles will not fuse with the presynaptic membrane. If the vesicles do not fuse, then it means that there will be no acetylcholine that is released into the synaptic cleft, and that means that there would be no transmission of the pain. Okay, there will be no transmission of the pain impulse to the postsynaptic neuron. You only have to write 4 things of all of the things I've just said. All right, and obviously you can stretch out some of them. So if there is no binding of acetylcholine to the postsynaptic neuron, it means there will be no depolarization. So if you don't know that term, please go and watch action potentials and resting potentials on the channel. There will be no depolarization over here. And if there is no depolarization, it means that the impulse of pain would not be passed down to the brain. So you're basically blocking I'm just going to use, you're basically just blocking everything here from happening by the binding of endorphins to the endorphin receptor. I hope that makes sense. If it doesn't, again, you can just put it in the comments, let your colleagues assist you with their own responses or how they understand it. Suggest one disadvantage of using pharmaceutical drugs for reducing pain compared to using TENS. Well, this is quite easy. Pharmaceutical drugs can create a lot of dependency. I don't know if any of you have read about the opioid crisis in some parts of the US and also in some developing parts of the world where they're not really documented. But usually you would find that in places where people use painkillers, some people become really dependent and are unable to function until they have taken a really strong painkiller. So it does create dependency, and that is a big disadvantage of a pharmaceutical drug. It would be a lot better to use an electrical impulse to just stimulate the nerves around the area so that they can become active. Let's look at number 6. So number 6 is about homeostasis. Number 5 was about coordination. So this is a chapter 14 question over here. Thermoregulation is the control of the core temperature of the body. Thermoreceptors send information about a change in core temperature to the hypothalamus. The hypothalamus controls the core temperature by sending impulses to activate several physiological responses. Table 6.1 lists physiological responses to a decrease. So take note, it says a decrease in core temperature. Then you're asked to complete this table, and I think it was for 4 marks in total. So 1 mark for each section that you get correctly. So here, if there's a decrease in core temperature, what that means is that the weather has become cooler. All right. So that cooling of the weather can result in you doing certain things. For example, when it's cold, what do you do as a person? Some of you start to shiver. Some people speak in a different language. Ugh. That's because it's cold, okay? So these are some of the physiological responses. What you're being asked to do in this question is explain what these responses are achieving. So over here, for example, it says blood vessels near the surface of the skin constrict, which means they become smaller or they become narrow. What this means, the explanation is that the body will lose less heat. Okay, so lose less. I'm just going to write less as an arrow coming down. That obviously doesn't look like an arrow, but yeah, lose less heat. That's why they contract, okay, or they constrict. Then shivering occurs. So obviously whenever you're shivering, what's actually happening is that your skeletal muscles are involuntarily shaking, if that makes sense. So it's like a muscle contraction of some sort, and it generates heat. Okay, so that would generate heat. Then your hairs rise. Obviously, you've noticed when people are cold that the hairs on their skin will stand. And what this means is that the hair will trap heat closer to the skin. Okay, so the trap heat closer to the skin or trap air closer to the skin, and that air will then form some sort of insulation against whatever is happening on the outside. And over here, reduction in the production of sweat. So that's to decrease heat loss. So decrease loss of heat from the body. So this is what happens whenever you're cold, and these are the explanations for why they happen. Let's look at B. Animals can also use behavior to adapt to temperature changes. Suggest why some animals curl up their bodies in cold weather. Well, that's pretty easy. Why do you curl up when it's cold? For example, when you lie in your bed and it's cold, why do you push your knees together and bring them close to your chest? It's simply because you are trying to reduce your surface area so that you reduce so you reduce your heat loss. That's typically the answer, or that's really the answer to that question. Let's look at C. The Sitatunga there, I'm not going to say its name, that name, and the Nile monitor lizard live in Africa. So that's the deer and that's the lizard. By the way, when you're working on paper, these pictures are a lot clearer. So if you can't see them on this video, please do not despair. And also, they're not going to tell you to be like, oh, look at the picture and tell us how many horns does it have, or that's not typically what the question would ask you, so it doesn't really matter. An investigation was carried out to measure the core temperature of the Sitatuga and the Nile monitor as the environmental temperature was increased. The results are shown in figure 6.3. So now let's look at this. This is the deer and this is the lizard. Okay, and you can see here temperature started increasing. So I'm guessing this was from around, let's say, if this is 10, so let's say this was around 5 degrees. And as we increased the temperature, you can see that the temperature of the Nile monitor lizard continued to increase, while the temperature of the Sitatunga sort of stayed within a specific range. If you just look up here where I'm circling, so it stayed within a specific range. And then they say describe the result. So describe is the verb that's used here. They're not asking you to explain it. Okay, so describe means look at what the graph is showing and just tell us what trend it is. Put it in words. First things first, like I said, the Sitatunga has a fairly constant temperature compared to the Nile monitor. Okay, the Nile monitor's body temperature increases as the environmental temperature increases. So those are two things that you can say there. And then you can say, for example, just maybe pick like the units and say like at 40 degrees, for example, environmental temperature, the Nile monitor was at, I guess this is maybe around 37, while the Sitatunga was somewhere around 38. So those are some of the things that you can say. And obviously remember to put the units. Please don't just put numbers. Don't just say, oh, 38. Make sure you write 38 degrees Celsius so that you can get full marks, because sometimes you might lose marks for not doing that. Then in 2, it says when the environmental temperature is low, now monitors are relatively inactive. Suggest one disadvantage to the now monitor of being inactive. Well, if the now monitor is inactive, remember it's a lizard, it can be easily eaten by a predator. So that would be one big disadvantage. If it's inactive, it also means that it is unable to go out and look for food because it is inactive. So that would be a good answer for you to give there. Thermoregulation relies on negative feedback. Explain what is meant by negative feedback. So this is usually what homeostasis starts with. So if you haven't watched the videos on homeostasis, it's a good time to go and do so. Negative feedback simply means that you are restoring a change in a physiological term or a physiological event. You're restoring it back to a set value or close to an ideal value. So you can also say that negative feedback is more or less reversing the effect of something. So for example, if the temperature gets too cold, your body tries to raise its temperature. If the temperature gets too warm, your body tries to cool you off. So negative feedback in this case, you would say, is a process whereby a change in a physiological factor is set back to an ideal value or set back close to an ideal value in order to ensure that it stays at a set point. In other words, it is used to counteract physiological change and to restore things to an ideal point. Okay, let's look at number 7. Number 7 is inherited change. This is chapter 16 in the textbook. It's one of my favorite chapters. Let's look at what they ask you here. People vary in which molecules are present on the surface of their red blood cells. For example, in the ABO blood group system, people may be categorized into one of 4 blood groups. So you can either be A, B, AB, or O. A single gene, which is I, with 3 alleles determine these blood groups. Gene I is an example of a gene with multiple alleles. Alleles IA and IB are codominant, which is why you can have AB. Allele IO is recessive to alleles IA and IB. Explain what is meant by codominant and recessive. Codominant means that you will have 2 dominant alleles, so both alleles are dominant and they can both be expressed in a heterozygous individual. So remember, like with dominance and recessive, usually it says IO is recessive to IA. So it means that for someone who is blood group A, they can either be this, which is dominant recessive and is heterozygous, or they can be this, which is homozygous dominant. But in this case for A and B, they are codominant. So even though the person is heterozygous, both of these alleles will be seen, which is why they are AB, and this person is A, sorry. So I hope that makes sense. Please watch the videos on inherited change. I don't want to spend too much time going over it. Okay, so what is meant by codominant? Codominant means that you will see both alleles expressed in a heterozygous individual. Okay, recessive means that the allele is only expressed in a homozygous individual and when there is no dominant allele present. Okay, so for you to see a recessive trait, there has to be 0 dominant alleles, and you can only see recessive if there, 0 dominant alleles, which basically means the organism is homozygous recessive. Okay, table 7.1, let's fill this table in. Shows a cross between a person with blood group A and a person with blood group B. And then it says complete the table. This is nice and fun. So first things first, this is person A, they are blood group A, but their genotype is AO. Remember that O is recessive, so this person is a heterozygous, okay? This is also a heterozygous person. They are BO, okay? The gametes in this case would be IA, okay, and IO. And over here it would be IB and IO. All right, and then they say the offspring genotype. So in this case, you might be confused if you're looking at the table over here. So I always say just take your pencil, get on the question paper somewhere in the corner or on a blank page on the question paper and do this. They've told you that these are the guys involved. Okay, that's for parent number 1. Then you draw a Punnett square, and parent number 2 is IB, IO. Okay, so now let's see here. This would be IA, IB. Okay, remember you're crossing what's on top here. I'm just going to use a black ink. You're crossing what's on top here with what's by the side here. So that's IA, IB. This will be IB, IO. And then over here it's IA, IO. And here it's IO, IO. All right. So now they say offspring genotype. Obviously, they're not expecting you to write it in any particular order, but you can simply just pick out these are the offspring here in these 4 squares. So here you can just come and then put in what you've got. IA, IB. Always remember the genotype will contain the 2 different alleles. Gametes only have the single allele standing by themselves, all right? And then over here you would have IB, IO, then it's IA, IO, and then it's IO, IO. Then they say, what's the offspring phenotype? So in this case, they're asking you, what would the blood group of the person be? Because A and B are codominant, this would be AB. Okay, B is dominant to O, so this would be B. A is dominant to O, that would be A. And O is recessive and it's homozygous over there, so that would be O. And that would be your answer. That's it. Very easy. Then the last question for that one, it says hemophilia is a blood condition where bleeding continues for a long period after a blood vessel has been damaged. A person with hemophilia has a mutation in a gene on the X chromosome, which results in the lack of a blood clotting factor. Explain why a man who has hemophilia cannot pass the condition to his male offspring. I love that they asked this question because I explained it in the videos on inherited change. So again, please check those out. But over here, if a man has hemophilia, let's do the example. This hemophilia, they say, is linked to a mutation on the X chromosome. So you'd usually write it as that, and then Y. Okay. Let's assume that his wife is normal. Obviously, in this case, it doesn't really matter because we are dealing with a son. If we were dealing with a daughter, the wife's genotype would be important. So this man, if he were to have a daughter, he would pass on his hemophilia, not his hemophilia. He would pass on the hemophilia to his daughter. However, because hemophilia doesn't sit on the Y chromosome, if he had a son, the son would not get it. Because remember, when an offspring is being formed, if it's going to be a male child, the man donates the Y chromosome. If it's going to be female, he donates the X chromosome. So what this simply means is that this man is able to pass hemophilia to his daughter, but the question asks you, why can't he pass it to his male offspring, so his son? So again, you then speak about the fact that there's only one X chromosome in a male child, and that X chromosome comes from the mother. Okay, the father only passes the Y chromosome to his son. The Y chromosome does not carry the hemophilia mutation, and as a result of that, the son cannot get hemophilia. Very simple and straightforward. Okay, now we're getting very close to the end. Let's see what it says here. Humans use selective breeding to improve the features of crop plants and domestic animals. State another term for selective breeding. So another term for selective breeding is artificial selection, because what you're basically doing is you are choosing what traits you want to happen. You're not letting things take their natural course. You're not saying, well, I'll just leave the plants and then see what happens. In this case, you're like, I want to make sure that this is what I get out of these plants breeding. And I can see the second question is already sort of alluding to that. So it says, describe how selective breeding can be used to improve the milk yield of dairy cattle. In this case, what that means is that what you do, or more or less, is that you would maybe pick a female cow that produces a lot of milk. Okay, so you start the female cow that produces a lot of milk. Please note the emphasis on female. You can't produce milk from a bull. But what you can find in a bull is that it might have certain traits that also stimulate milk production because its mother produced a lot of milk. So you can then say you choose a female cow that produces a lot of milk, and you choose a bull or a male cow, whatever it is that you might call it, with a generation of females that produce a lot of milk. You cross the two together, and then you take their offspring and you start to breed their offspring with each other. What this will do is that it will increase the propensity for the high milk yield gene to be passed on from offspring to offspring to offspring, and as a result of that, you will then be able to have a lot of cattle that produce a lot of milk. So you pass on the desirable traits. Something else that you can do in terms of selective breeding is you can use artificial insemination, whereby you can take maybe the sperm from the bull rather, whose mothers have a high milk yield, and then you put that into the female that has high milk yield, and as a result of that, you're able to produce offspring that would then give you a high milk yield. So that's how selective breeding would typically happen. And I would just want to say that if that flew over your head, just go and watch the videos on selection and evolution. That's, I think, chapter 17, if I'm not mistaken. So please make sure that you give that a look as well. Explain what is meant by disruptive selection. So whenever you're doing selection, or when you get to the selection chapter, you will see that there are different types of selection. There's directional selection, there's disruptive selection, and all of that. Disruptive selection is the type of selection that sort of selects against the intermediate groups. So the graph, I think, looks something like that. All right, so let's say, for example, these are really short people and these are really tall people. What disruptive selection will do is that it will attack the people. There'll be a selection pressure against the people who are of average height. All right, so it would maybe say people who are below 5 feet, between 3 feet and 5 feet 4, those people will be selected against. And so if those people are selected against, then maybe there's something that just kills them for some reason. So that's what disruptive selection does. And I know that that's not a great example, especially when you're talking about people. People might be like, oh my God, why would you say that? But disruptive selection is simply selection against the intermediate groups in the population, allowing for only the two extremes of the population to survive. All right, so if only the two extremes survive, then things change. So again, it might even be in terms of body weight. So maybe like a very skinny cat might not be desirable to a predator, and a very fat cat might also not be desirable. Maybe the predator just likes an average sized cat. So the average sized cats will then get eaten up by the predator, leaving the really fat cats and the really skinny cats to survive. So that is what disruptive selection does. If you want to get more detail, watch the videos on selection, please. And now to the last part of this paper. So again, like I said, paper 4 is your longest paper, and it is for 100 marks. So it's a paper you don't want to take lightly at all. You want to make sure you give it your very, very best. When you get to section 2, which is section B in the paper, you will see that it says answer one question. For some reason, I have seen students who just did not understand that they had to read the instructions and proceeded to take time to answer both questions in section B. Answering both questions in section B will not give you more marks because only one question will be marked, and it will usually be the first one. Nobody is going to say, oh, I think you did well on the second one more than the first one, so let me mark the first one. So always answer only one question. Each question is 15 marks, and you have a choice. You can decide for yourself, okay, well, which topic am I most comfortable with? I need to say this, that you can't answer question 9A and 10B. That's not how it works. If you're going to answer question 9, you must answer 9A and B. If you're answering question 10, you must answer 10A and B. So please don't go to the exam and be like, I'm going to do a mix and match. I know 9B and 10A, so I will answer those two. You will not get marks for that. Okay? So let's look at these two questions in a bit more detail. The first question says, describe how you would carry out an investigation to calculate the effect of substrate concentration on the rate of respiration of yeast in anaerobic conditions using a redox indicator such as DCPIP or methylene blue, and that is for 8 marks. And the second question is compare respiration in anaerobic conditions in mammalian tissue and in yeast cells. From reading this question, I can already tell that my students who wrote in May, June 2021 probably skipped this one and figured, well, there is no way I am going to do this, because this is more or less like a paper 5. They're asking you to design an experiment. So first things first, you need to ask yourself, well, we are dealing with what? Respiration, anaerobic conditions, yeast. And we're also doing an indicator such as the CPIP. What does yeast use to respire under anaerobic conditions? The substrate is glucose. Okay. So again, remember you're dealing with respiration, so it's glucose. Or you can even say fructose, doesn't matter. So if it's glucose, then it means that, and it says calculate the effect of substrate concentration. If you're calculating the effect of substrate concentration, it means you must change the concentration of the substrate. Okay, so you need to have different concentrations of glucose. So I'm just going to write it there. You have different concentrations of glucose, and you can write this out. I prefer for students to write in outlines, but if you're unable to write in outlines, again, you can just write it, but be as clear as possible. So I would say prepare varying concentrations of glucose in beakers, for example. Then add the same volume of yeast. So yeast has to be constant because you're not investigating the effect of yeast. All right, the yeast has to be constant. So the reason why yeast is constant is that you're saying if I put 2 grams of yeast in 10 microliters, not 10 microliters. If I put 2 grams of yeast in a 1 molar solution of glucose, and I put 2 grams of yeast in a 0.5 molar solution of glucose, what would the impact be? In that case, you are varying the concentration of glucose. So glucose is the one having the impact, okay, on the yeast. So same yeast, same amount of yeast. Obviously, you also want the volume to stay the same. Okay, so volume stays the same. And also in this case, you also want to then add your redox indicator, and it has to be the same amount. So again, because you're varying glucose, you don't want anything else to vary so that your results will be reliable. So also it would be the same volume of indicator. So I'm just going to write it as indicator or indic same. This already is 4 marks, just by the way. All right. Then obviously you want it to be at a constant temperature. So you put it at temperature, stays the same. This is you designing an experiment. So if you've been practicing paper 5, I'm sure this makes a lot of sense to you. If you haven't, please don't overlook paper 5. It can stump you in ways that you did not imagine. So make sure you give it a good shot. All right. So temperature also stays the same. And obviously, because you're dealing in anaerobic conditions, you also want to create anaerobic. So you need to create an anaerobic condition. The way to create an anaerobic condition in the lab is usually to put a layer of oil on top of your experiment, because what the oil will do is it will prevent oxygen from dissolving in the solution or even penetrating the solution, so that way you create an anaerobic environment. And then in this case you can then time the experiment, so you set a time and see what happens with the change of color. So the DCPIP, it says it's a redox indicator. Redox indicators usually change color. All right. So then you can say time experiment to see so time experiment to check for color change. Okay, so change in color. And let's see how many have we written now. We've written 1, 2, 3, 4, 5, 6, 7, and you only need to write 8, okay, because it's just 8 marks, so 8 is fine. And then you can then say over here that you repeat the experiment in triplicate, so you maybe repeat 3 times and calculate mean value. All right, so that would then give you 8 marks, and that's all they're asking from you in this question. That's it. Then in the second question, it says compare respiration in anaerobic conditions in mammalian tissue and in yeast cells. So first things first, it says compare. So whenever they say compare, they're asking you to show both the similarities and the differences. That's what compare means. They're not saying differentiate. Differentiate means show the differences. Compare means what are the similarities and what are the differences. So again, in both mammalian tissue, so that's in things like your human cells, and in yeast cells, you have anaerobic respiration happening in the cytoplasm. Glycolysis will always happen. Please, if you haven't watched the videos on respiration, please make sure you check them out. Glycolysis will happen. The amount of CO2 you generate only comes from glycolysis, and that is 2 ATP. Well, just know that the amount of ATP, I feel like I mentioned a completely different chemical just now. The amount of ATP you generate would be just 2 ATP because you only undergo glycolysis. And then you can also speak of the fact, actually, let's just write it down. So they both happen in the cytoplasm. I'm not going to write everything out. They both happen in the cytoplasm. Glycolysis is where they both start. 2 ATP is generated. Okay, so 2 ATP is generated. You also need to mention here that the final acceptor, the final hydrogen acceptor in mammalian tissue is pyruvate. Okay, so pyruvate is the final hydrogen acceptor in mammalian tissue, whereas in yeast it is ethanol. Okay, then in mammalian tissue the final product is lactate or lactic acid, whereas in yeast it is ethanol. Okay, in mammalian tissue, because it's lactate, the enzyme involved is called lactate dehydrogenase. I'm not going to write that out. I'm just going to write dehyde, but it's a dehydrogenase. Whereas in yeast tissue, it is ethanol dehydrogenase. Okay. And for the last one, because it's 7 marks, so you only need to write 7 things. In mammalian tissue, the lactic acid fermentation is a reversible process, whereas in the yeast tissue, it's not. So when yeasts make alcohol, they don't then take that alcohol and make it back into pyruvate or into ethanol or anything like that. It stays as ethanol, which is why you can have wine and you can have bread taste the way it does. The bread just doesn't wake up and be like, oh, I no longer feel like having ethanol in me, so I'm just going to become something else. That's not what happens, all right? So those would give you full marks for this question. Let's do the second one. So again, remember that you only have to answer either question 9 or question 10. Please do not answer both questions, all right? Make sure you pick the one you're most comfortable with. And if I had to take a guess, I am guessing most of my students maybe chose, well, I know they didn't like speciation and so on, so I'm guessing maybe they chose the previous one, but I also hope that they chose this one because I think this one's quite easy. So yeah, so question 10A, explain how speciation can occur due to geographical separation. So in this case, you will say, I'm just going to write it here. First of all, this type of separation is, you can just start by saying geographical separation leads to allopatric speciation. That's what the technical term is, allopatric speciation. All right. Now, what results in this is that there's a geographical barrier. So a geographical barrier exists between two groups of a population. All right, so it's the same population at first, but then something separates them. So it could be maybe something like a river, it could be a valley, anything at all, but they start off as one species. As a result of that, because of this geographical barrier, the guys on the one side will be the ones mating with each other, and the guys on the other side will be mating with each other. So it means that mating is restricted between the two groups. So mating restricted. Okay, if mating is restricted, it means that there is no gene flow between the two groups. All right. If there is no gene flow between the two groups, what you will find is that different mutations will start to occur. So depending on where they are. So let's assume, for example, there is a vast piece of land. So I'm just going to try to draw something here. This is a vast piece of land, but for some weird reason, some river starts flowing through this land, and eventually it becomes like a river pathway. So initially, all the organisms that lived, okay, can't do that. So let's rather do it this way. So I'm just going to write this as A, and this will be B. So let's assume that initially all the individuals who lived here did not have this vast piece of land that formed between them. Okay, they were all just jolly happy people, but now there's a river that's flowing in between, so it means they can't cross over to the other side. Let's assume this is a really deep river. They can't cross to the other side to mate with the guys over here, so they start mating with each other only, and these guys also start mating with each other. Now let's say on part B there are lots of trees, right? Whereas on part A it's maybe just flat ground. The way these guys will evolve will become different from the way the guys on section A will evolve. And that is simply because, for example, if there are a lot of trees, maybe the people who are on B will then learn how to climb trees, maybe for the sake of getting fruit or anything like that. Whereas the guys on land A, because the ground is flat and there are no trees, they don't need to learn how to climb. They will then, you know, flow differently. So no gene flow, and then mutations also will occur differently. Mutations will be different. Obviously, because of this, after a while, the way of life of the guys on section A will be different from the guys on section B, and what you will find is what would be then founder's effect. Not only just the way of life, even the genetic makeup. Because remember, if these guys are only mating with each other, then only certain traits are being passed on, and these guys are also passing only certain traits on. So you'd have founder's effect or genetic drift happening. How many is this? This is 1, 2, 3, 4, 5, 6. We need to write 2 more things. Also, you can then say that they would no longer be able to breed with each other because they've evolved differently. Due to this geographical barrier, they've basically followed very different lives, and as a result of that, they will no longer be able to mate with each other, so can no longer mate. Well, I'm not going to write that out, but you get the gist. They can no longer mate with each other. And another thing that you might be able to say in this case is that the gene pool will be different, okay? Because there is no gene flow, obviously the gene pool will occur differently. So those 8 things will give you full marks here. And then for the last one, it says explain using examples why species may become extinct. In case you just want to say that if you watch this section now and you're like, what just happened? Please go watch the video I did on selection. It's right there on the on the page, on the playlist. I think it's chapter 17. So just watch about allopatric speciation and so on. So why would species become extinct? There are so many reasons. They can't adapt. There's climate change that's happening. Sometimes there's competition. There are diseases that cause them to be wiped out. You can have habitat loss. Simply just think of all the things that are happening in the environment right now that are displacing many of the species that we should be caring about. And if you're able to write 7 of those, then you should have full answers. So again, the 7 things that come to mind, I don't know if I can think of 7, but climate change definitely is number 1. Environmental loss or habitat loss is another one. That's one of the reasons why species become extinct. Competition for food, diseases, predation, those are all things that put species at risk as well. And also you can even add human activities such as hunting. I think those are 6 things I've just mentioned, and if you can think of a 7th one, then please add it to that, and that would give you full marks for that answer. So this is the end of paper 4, and I hope that you have found this helpful for those of you who are preparing. The last paper I will do will be paper 5, and afterwards I will be going on a very long break because I have to do other things that have to do with school, but I will post occasionally to answer questions and also to help you with some of your revision for concepts. So again, please make sure you study all the chapters that you should know for each exam. For paper 4, it's chapters 12 to 19. For paper 5, it's the same. For papers 1, 2, and 3, you should study from chapters 1 to chapter 11. Paper 3 is a laboratory-based paper, so it's more about carrying out experiments, which you should practice with your teacher in school. All right then, until the next video, have a good time, guys. Goodbye.